So let's start our next session. So this session is about LLM-based ontology mapping. The first talk will be given by George Weiguo Cao from Mayo Clinic about the ontology-based LLM for clinical trial classification and the vaccine study. Go ahead.
Okay, and this study is conducted in collaboration with UW, Mayo Clinic, Yifeng, and UT Healthy. And the title is Ontology-Guided Prompting Improved Large Language Model Classification of Clinical Trial Phases and Myocarditis Signals in Moderna Vaccine Studies. The motivation comes from the first is the rare myocarditis cases were emerged after vaccine, COVID-19 vaccine, especially from the Moderna. And the second is the CT report. It is about 40 to 55 myocarditis cases per million second dose in young males. The third is the pre-clinical phase 1 to 3 trials reported no myocarditis cases. So there are the two key questions. The first is how can we reconcile the absence of such findings in clinical trial with clear signals detected in post-marketing surveillance. And the second is can we use ontology-guided large language model to close this gap? So there is we noticed several problems from the clinical trial and the virus, and based on the problems we find three gaps. The first gap is the terminology gap. The second is the time window gap, or you call it is also called scale gap. The third is the feedback gap. So to tackle these issues, so there is three objectives. The first is ontology-guided large language model extraction from clinical trial. So the purpose is to detect the Moderna vaccine and identify the myocarditis information, and then extract the trial phases 1 to 4. And the second is a VAERS-based surveillance analysis. And its purpose is to quantify the real-world myocarditis signal after Moderna vaccines and characterize the age, sex, timing, and outcomes. The third is the signal to surveillance loop integration, and this it will define a reusable signal card on the trial side and translate it to surveillance queries on the website and feedback to the prompt and the trial design. And for the data, we include the came from several datasets. The first is the clinical trial dataset. We collect 180 trials in total, including 55 Moderna trials. And the second dataset is the virus, and we obtained 1,014 Moderna-linked cases. The third is 3 ontologies, including the vaccine ontology, the ontology of adverse event, and the human phenotype ontologies. So to solve this problem, and we propose this method. This method is a loop architecture, and from the surveillance signal detection to prospective signal mining. And the third step is the gap analysis and the knowledge generation. And from with the knowledge and the analysis, then there is some feedback and refinement. And this feedback and remind is we use this the feedback and remind, and then we we improve the prompt and for the next cycle. So we will report those steps one by one. The first step is the surveillance signal detection. And its purpose is to use the virus and other post-marketing system to detect the potential safety signals, such as the myocarditis and after Moderna vaccines. And second is, and then characterize the basic patterns. The second step is retrospective signal mining, and we use the signal detected from the virus and go back to the clinical trial to use the ontology-guided large language model prompt to respectively mine the trials. And the third is the gap analysis and the knowledge generation. We contrast the endpoint evaluated in the clinical trial with the real-world signal identified from the surveillance, and then identify the gaps in phase and sample size and the eligibility criteria and the window and AE definitions. Summarize the findings as a structured knowledge for the myocarditis. The fourth step, last step, is to use the gap analysis to refine the ontology and the prompt and translate this refined knowledge to the recommendation that informs the future trial design, and close the loop by feeding this refinement back to the next cycle. And use this method and this data, and we did some experiment. And from the experiment, we could say, and we list the result in this table, and from there is totally there is four phases. Because there is for some of the articles they did not mention the phases. We just label it NA. From the table, we could say the accuracy was increased from 74 to 95, and especially for the AA trials, there totally there is 56. The original method is about is less than 20%, and after our ontology-guided method, its accuracy will improve to 100. And the slight decline was the earlier phase. Earlier phase because there is only 2 trials. The trial number is 2, so I think it is the rare cases. And the second decline is the phase 4. But phase 4, I think it is also due to the rare cases. So it dropped about 80%. And we also compare the all result with the baseline. The baseline is the original method. It is a GPT prompt without the ontology. And this is the computer metrics. And among the 4 stages, because there is NA, actually there is 4. The earlier, the phase 1, phase 2, phase 3, phase 4, phase 5, and the NA. And the left is the method without the ontology knowledge, and the right is the ontology-based guidance. And from the second line is NA, you could say for in the original image or original method, and so there is totally there is 56 and 43 error, and with all the ontology large model, they are all corrected. And the decline you could say from the last line, the phase 4. And for the baseline, they are all correct. But for the ontology enhanced large language model, 7 was the error. So this is maybe this is our future works. And for the second experiment is we draw the curves, and we will collect data from 2021, the January 2021 to this year, and we collect the data and plot the curve. So totally there is 1,040 Moderna cases, and the sharp signal in it should be the median 2021. You could say this is the peak, and there is a clear decline after 2022, and this result is consistent with the CDC estimation. And the last. Except the comparison and the trend, we also calculate the age and the demographics, and calculate and this is the distributions. And for the from 2021 to 2024, yeah, the among the cases about the male is about 82%, and 85% within 7 days after the second dose, and for the 2021 there is a strong concentration. Yeah, you could say the major people there. Yeah, this is the peak, and happens for the adolescents and the young adults. The age ranged from 18 about to 40. And for the 2022 to 2024, the distribution become broader. And yeah, as a conclusion, the ontology-guided prompt framework show great potential power for our study, and it inject the ontology knowledge into the GPT-4 and boost the phase classifications. And the second to surveillance loop algorithm defines and explicit reusable patterns. The third is the various myocarditis characterization define confirm the young male dose, second dose signal with the real world timing, demographics, and outcome data. And the last conclusion is the generalization paradigm can extend to other vaccines, drugs, adverse events, and reporting system. There's some limitations, and the future works. And in the future, we will expand this method to other vaccine platforms and explore fine-tuning to augment the larger model to further reduce the misclassifications, for example, especially for the fitful. And we automate the signal class generation and integrate the regular workflows. This is a reference. Thank you.
Okay, great. We are out of time, but any quick question if anyone has one? So no, okay, great. Thank you, George, for your good talk.